Good evening to everybody. I am Dr. Stabilisers, Director of Maliba Pharmacy College and Dean PG and Research of Kolkata University. Uh, as a part of YouTube, we are organizing number of the webinar in this lockdown period. In last few weeks before, we have a successful research methodology webinar, and in the next week we have a faculty development program on the live in resources, so you can register also. Today we have another faculty member that we organize this webinar. That is a YouTube practice that we are identified the topics which are interested for the student as well as the researcher, and we are identified the speaker or faculty is a master in that. And in that way, today we have a webinar on COVID-19 outbreak and possible formulation options for approved and new drugs. The speaker is Yogeshwar Bachhav, Dr. Yogeshwar Bachhav, presently working as an Associate Director, Pharmacy Department, consultant at iCuris Anti-Infective Cures, Germany. He is there. He did his B.Pharm from North Maharashtra University, and he is a ranker, third ranker in the university, and did his M.Pharm in pharmacochemistry from the University, Bombay University, or we say Chemical Institute of Technology. He stood first in M.Pharm and also did his PhD from the same institute. He's published 16 international research paper with a good impact factor on his name, 2 patents, Indian patents, and 6 international patent are there. And he published 30 conference proceedings and edited book in the Ville VCH, invited as a speaker and number of international seminars and conferences. He is starting his career from the Sun Pharma Bombay, then further proceed to the Pantas Biosolutions, then Formulation Development Manager at the Deep Bounder in Switzerland, and at present he is working in Germany. Today, and he's published his expertise in CMC and dossier preparations and other. So today we have very learned faculty member or speaker. So now I invite Dr. Yogeshwar to start his speech and enlighten us. Thank you.
Okay. Good afternoon, all. I'd like to thank Professor Shailesh Shah, Professor Pranav Shah, for giving me this opportunity to speak at this event, and it's my pleasure. So, topic of today's talk is COVID-19 outbreak, possible formulation options for repurposing or new drugs under development. So basically, I just tried to summarize a little bit about COVID-19 outbreak, then what are the options we do have available from repurposed drugs, what is the status of the vaccine research, how the vaccines basically work, and when we have like new drug or vaccine, what are the formulation challenges we face. So let's start with COVID-19 outbreak. So coronaviruses are the family of viruses, and they are mainly coming from the animals moving to the, let's say, humans. And the first symptoms you see, these are like cold-like symptoms. So COVID-19 is very related to the virus we heard before, maybe in 2002, was a SARS virus. Then in 2012, we heard about MERS virus, which is a Middle East respiratory symptoms. That was not very severe. We had like 2,500 cases globally with 900 days. So this COVID-19 is little different from these two viruses because spectrum of disease is quite broad. So 80% cases there are like mild infections, and 20% you have severe infection. And currently the death rate is varying between 0.7 to 3.4%, but that also depends on the location, because the location is also linked to the access to good hospital care. And recently, the Chinese scientists were publishing that the COVID-19 has mutated into two strains. So one is more aggressive than other, and this makes a vaccine development little difficult. So basically, how this coronavirus is spreading and how we can protect from this virus. So once the
Yogesh sir,
person
if
is
hello, Yogesh sir, if you can speak a bit louder,
okay,
please. Sorry to interrupt you.
no
Thanks.
problem. Can you hear me now?
Yeah.
Okay,
Thanks.
no problem. So basically, virus is spreading in a similar way as the virus from flu or common cold is spreading. So it's coming into form of large droplets produced by coughs and sneezes, and these germs can live on the surface for a few hours. So health officials basically try to predict, or at least they indicate, that the chances of infection are quite high when we are in contact with the carrier person, at least within the diameter of 2 meters, and if we are there for 15 minutes. And to stop this spread of the virus, normally they recommend catch it, bin it, and kill it approach. In that case, what one has to do, we just have to catch the sneezes, coughs into the tissues and throwing them away. And the patient should wash their hands very carefully. And for the normal person as well as the infected people, it's very important they should regularly wash their hands, so that the chances of contamination, they are very less. So little bit about this outbreak globally. As you can see, and this data is from a study. So globally, we had around 5.1 million cases. 2.8 they are active, 1.9 are recovered, and around 3,32,000 deaths are there. So US is still on the top from this chart, followed by Russia, Brazil, other European countries. And when we see about India, we have like 1,18,000 cases. There are still 66 active cases, around 40,000 recovered, and 3,500 deaths. So, if you see the numbers, India has been still able to control this disease, quite better way compared to the Western countries. But again, as you all know, the graph is not coming down, and number of cases are still increasing. So certainly we need a treatment for this disease. And when you go see for the treatment options for this coronavirus infections, maybe we need a new drug, small molecule, maybe vaccines, or maybe we have option of repurposing of existing drug. And when you think about repurposing, what options we do have? So recently there was a lot of hype around hydroxychloroquine when there was a shipment requests coming from Western countries to Indian government. That's where we saw maybe hydroxychloroquine is very useful drug, and even in India, if you see, it is regularly being used as a prophylactic treatment for healthcare workers. Other options we have like remdesivir, favipiravir, or maybe combinations. So maybe repurposing option could be desired option. Why? Because in this pandemic, the time is very crucial, and if you need drug is approved, you just need to reformulate if needed, and that is readily available for the treatment. But if you see all the clinical data around or the published report, it does not look like if the repurposing option is still working. Maybe some more studies are needed. We'll discuss that on. On the contrary, if we are able to develop a new drug or vaccine, it is a time consuming approach, but it is certainly going to be very precise because we are specially developing this drug for this against this specific virus. So on this slide, I just tried to summarize the drugs in pipeline for COVID-19, and you see the mechanism of action, the chemical classes. So like chloroquine, hydroxychloroquine. So basically, this is act by changing the pH of endosomes, and that's how they prevent the viral entry. So these drugs act in vitro quite nicely against the virus, but in vivo mechanism cannot be established. Then you have other antiviral drugs like remdesivir, ribavirin, lopinavir, darunavir. Most of them they are again already established against, let's say, HIV virus or the influenza virus, and they just act by inhibiting the viral RNA synthesis. So still clinical studies are ongoing to prove if these drugs can really act against coronavirus. You also have some drugs being evaluated clinically from the class of interferons, and they basically act by modulating the immunity, and that's how they try to block this viral infection. Some antibodies are also being evaluated for this infection. The data is still awaited, but having said that, a lot of studies currently ongoing to check if we have any options for repurposing. The remdesivir is very interesting example. I tried to get some data published by a French journal, and on the right of this slide, you can see they tried to determine the viral shedding by quantitative PCR from the samples from upper and lower respiratory tract, and you will see that study was done for 28 days, and you can see that compared to control samples, the remdesivir was not really performed very well, and that clearly indicates the efficacy of the drug against the viral infection. On the left, I just tried to copy the protocol they have used when they tried to screen 255 participants. Out of this, the study was done like 237 patients. So this gives little bit idea how one can run the clinical trials for any new drug or repurposed drug on the corona patients. So let's say remdesivir is very efficacious. So what are the chances of availability of this drug in India? So this drug belongs to U.S. company Gilead, and Indian patent was granted for this particular drug in Feb this year. So when any pharma company wishes to use this drug in India, of course they need to discuss with Gilead. And recently, the U.S. agencies they came up with the data that this drug can shorten the recovery from 15 to 11 days, and even the death rate improvement was noted from 11.6% to 8%. Important to note that this drug is administered by IV route, so certainly it has to be used only under hospital settings. On this slide, you see more data regarding this clinical outcome. During the initial studies, in fact, they did not notice any clinically significant difference between the control group and remdesivir group, and the data was not that promising. So even they commented that some combination studies are needed, maybe dose needs to be increased, and it has to be even tested in the earlier let's say corona infection cases. On the other hand, NIH reported some additional results when this drug was evaluated under ACCT screen, and there this study was done in fact on 1,063 patients, and those studies reported the patients who were receiving remdesivir they had like 31% faster recovery compared to control group, and even the time for recovery was 11 days for the drug arm compared to the placebo arm. So let's move to another drug, which is a favipiravir, how it acts. So on the top of this chart, you can see these are the viral particles. Basically, they are binding to the surface of the cells. They penetrate, uncoating takes place, then viral genetic material is released, and then it is using human cells machinery to produce more viral proteins. These viral proteins are coated, and then these viral particles are released from the human cells so that it can infect to the next healthy cell. So favipiravir, where it is, let's say, administered into the cells, it is undergoing phosphoribosylation, so that you have like favipiravir RMP. It goes under second cycle of phosphorylation, and at the end you have like favipiravir ribose triphosphate moiety. And basically, this triphosphate moiety is inhibiting the replication of viral proteins, and that's how it acts as antiviral drug. So, what is the outcome of favipiravir in terms of clinic and availability? So, if you see the clinical data, the clinical trial with favipiravir was done on 80 patients. Out of those, 35 got the drug treatment, rest 45 were on control arm. So, the median recovery time was 4 days for the drug arm compared to the placebo arm, where it was 11 days. And even the 7 days clinical recovery rate was increased to 55.8% to 71% in the drug arm, and that's how they tried to conclude the clinical efficacy of this drug. As you have seen regarding structure, it's a purine nucleic acid analogs. It acts by inhibiting the RNA polymerase. And if you would have seen in the news recently, Glenmark got a deal to have this drug for phase 3 trials, and even CSR has got the rights to use this drug for clinical investigation. So, and on this slide, you see the different trials ongoing globally using favipiravir. This drug is available in the form of tablets, and this is being used either alone or in combination with other drugs to check if this drug can work against this corona infection. And 2 days back, I was reading this news. Even the Corona Task Force for Mumbai was asking the Maharashtra government as well as Union government that combination of remdesivir and favipiravir should be made available quite urgently, because this could be uh, very helpful as a treatment option. So that also indicates the importance of these drugs. So, what about hydroxychloroquine? There's a lot of hype, as I said before. Maybe this drug works very well. There was a lot of global pressure, but if you see the pharmacological point of view, it doesn't really look like this drug works quite well in vitro. As I said before, it changes the endosomal pH. That's how it blocks the viral entry. But in vivo, this effect is not proven, and even clinically, you do not see it's working quite well. One of the French investigator did this trials in 36 patients, and they reported in fact drug is working quite well, but this trial was not randomized, and indicating that the investigator had a choice to choose the patient for drug arm and placebo arm, sorry. And that's the reason it was quite difficult to predict the data, and it was not that reliable. Even additional studies were done in Brazil using the chloroquine, but that trial has to be stopped because they reported some irregular heart-related adverse effect into the patient, and it has to be stopped. So one has to be very careful using this drug, especially for the patients having some cardiac risk. What about ongoing vaccine trials against corona? So maybe beginning of this week or last, you would have heard that U.S. biotech company Moderna, they revealed first data from the human trials. So the vaccine triggered an immune response in these phase 1 studies, and even the preclinical studies, they were able to protect mice from lung infections from coronavirus. And the overall, the reports in the press release were considered quite positive. But still, some scientists say that data is not yet complete, and therefore it's quite difficult to predict that the vaccine is working. So certainly, we need some more data. Then there were some additional reports were coming from the fast track could show that they were able to prevent the infection in lungs of monkeys, but not in other parts of body. So there is one trial ongoing from University of Oxford. In those trials, they were able to protect 6 monkeys from pneumonia, but still the animal noses were containing significant viral titer, and which was as good as the control group, indicating that lung infection they were able to control. But at the entry point, let's see, the nose tissues, the viral titer was still same. The Chinese group reported the similar incident, but still this data is quite important because it offers some clues how coronavirus vaccines generates the immune response, and this will help to come up with more promising candidates which could be moving to the next stage. Okay, so this is more about, like, say, the repurposing of the drug. But if nothing is working, certainly we need to think of the new drug development. So on this slide, you see more or less the summary of the new drug development. You start with the target discovery. When I say target, it's more receptor. You know which mode of action you need to block, which receptor we need to target. The target needs to be identified, then needs to be validated to make sure that once you hit that receptor, then some mechanism of action you are able to block. And that's once you know your pharmacophore, you start with heat generation. Heat generation tells you if that particular pharmacophore is binding to the receptor. Then you go with the lead identification. In that case, in lead identification, you try to check the structural details of that molecule by using the computer drug design, or maybe let's say SAR studies. And once you have sufficient data of the activity, you go for lead optimization. In that case, you need to check if the ADME of that particular compound is quite good, acceptable. If not, it has to be changed. And once you have all these properties, you move with animal models to check the efficacy and safety of that compound. GLP tox studies you need to do to make sure the drug is not toxic. You know the target human dose. You start with your clinical trials. And on this slide, you see the typical drafting of the clinical trials. So as you see, phase 1 studies, they are done on the healthy volunteers, sometimes even in patient, depending on the indication. This is done normally on 20 to 80 patient, runs from 1 to 2 years more or less. In this case, object is to check the safety and ADME properties of the drug interactions. Once you have the safety data of the drug, you go to phase 2 studies, which are typically proof of concept studies. The objective is to check the pharmacological activity of the drug. Normally, this is done on several hundred patients, run from 2 to 3 years. Once you have the positive phase 2 data, you go for phase 3 studies. Important to note here, not many small companies are able to do phase 3 studies because they are quite expensive. The risk of failure is very high. So mainly the big pharma companies are into phase 3 trials where the number of patients needed maybe in hundreds or thousands depending on the indication. They run for 3 to 4 years. And then you try to check the safety and efficacy of your drug. Then you also try to check the effect against placebo group to make sure your drug is really efficacious. And after phase 3 studies, it goes for filing. And after filing, the phase 4 studies, which are like post-operative trials, you need to continue. So, in summary, to develop any new drug, it needs at least 10 to 12 years with the investment of, let's say, 1 to $1.2 billion. So, one has to think under these pandemic conditions, if we have to develop a new drug, what resources, what amount of budget investment we need. Regarding the development of vaccines, so I came across very nice article in a Nature journal. So I tried to use some information from this journal. So currently there are 90 vaccines are being developed against corona from the research groups from the academia as well as pharma companies. And out of these, at least six groups, they already started their trials into the healthy human volunteers, which are typically phase 1 trials. And in parallel, they are also running some animal trials. So out of these vaccines, the maximum number comes from protein-based vaccines, followed by nucleic acid, viral vectors, and viruses. So, how do we develop immunity? We can see on this slide. So, basically, the viral particles, they are binding to the human cells. So, they are locking into this ACE2 receptor, which is present onto the cells. And once it is entering, then uncoating of this viral particle is taking place so that its genetic material is released. And then this basically this is RNA, and then it is using our cell's own machinery to for the, let's say, further replication. Then the further viral proteins are being produced, the coating takes place, and then it is followed by the release of the viral particles, so that it is impacting the next series of healthy cells. So how does the immunity work here really? So the antigen presenting cells, these are the part of our immune system. So they are detecting these viral particles. Basically, they are just engulfing these virus particles. They display the portion of these viral particles to activate the T helper cells, and T helper cells are the one which are triggering the immune responses. Then T cells also part of immune system. They make antibodies which can block the viruses so that they are not able to infect other cells, and they are also causing the destruction of viral particles. The cytotoxic T cells are identifying and destroying the, let's say, viral infected cells, and that's how the immunity really works. So if one has to develop any vaccine, they also need to take into account how let's say, the vaccine is going to trigger the immunity. Little bit about types of vaccine we discussed before. So it starts with weakened viruses. So in that case, the viral particles weakened by passing to the animal and human cells, so that some selective mutations are taking place, so that they are not viral and not causing the disease. Using this approach, Serum Institute Pune had a deal with a US based company. Then second class is inactivated viruses. So in this case, viral particles are treated with the chemicals or with heat treatment so that they are not active. We can also call them like attenuated viral particles. So they are not able to cause, they are not, let's say, virulent enough. They are not causing any disease, but they are able to create the immune response. Then you have nucleic acid based vaccines, so which are basically RNA and DNA based vaccines. So these nucleic acids are basically just inserted into the human cells, so that they are using the human machinery, human cellular machinery to produce the viral proteins which can then trigger the immune response. This looks quite simple technique compared to other vaccine categories you have seen, but still on the market there is no licensed vaccine using this approach so far. Next is a protein-based vaccine. So in that case, researchers just try to inject the coronavirus proteins. Maybe they are taken from the fragments of proteins or protein shells, which are more or less similar to coronavirus's outer coat. So these protein particles are still immunogenic. They can still trigger some immunity. Then you have viral vector vaccines. So in this class, what happens basically, they just take the viruses such as measles or adenoviruses, which are genetically modified, so that they are able to produce coronavirus like proteins into the body. And these proteins are still considered as immunogenic, and they are able to trigger the immunity. Then how we can develop a small molecules against corona, what should be the basis? And for that, it's very important to know the mechanism action for antiviral drugs. So basically, they act through four different mechanisms. In the first class, we have nucleoside analogs, and they interfere with the replication of viral genome. So the drugs from this class, they can only target the viruses which are having their own polymerase systems. So as just now we saw that coronavirus is not having its own polymerase assembly, so it's basically relying our human cell assembly, and that's the reason established drug like acyclovir or zerovudine, they are not effective against coronavirus. The second group, they are protease inhibitors, and these type of drugs are very active against HIV. So basically they are inhibiting the processing of viral proteins which are needed for final viral assembly and release. Third class of antivirals, they inhibit the step of viral uncoating. So if a virus is not uncoated, it is not able to release the genetic material into the human cells, and that's how they block the infection. The drugs such as amantadine, rimantadine, they belong to this class, which are approved against influenza. Then let's talk about fourth stage. So if you see the final stage of viral life cycle, it is the release of the matured viral particles from the host cells, and the drugs from fourth class, they are blocking this step. The drugs like Relenza and Tamiflu, they belong from this class and they are approved for influenza. So out of these four classes, or out of these four mechanisms, you need to consider one if you want to develop a small molecule against corona. I tried to take one case study from the literature. The Chinese scientists were trying to develop a new drug against corona. So in that case, they used the high throughput screening method to come up with the lead candidates from the commercial library of compounds. And the mechanism they targeted was cathepsin mediated glycoprotein processing. So basically, what happens? Cathepsin is a let's say enzyme present in the human cells, and this cathepsin is responsible for the processing of glycoproteins. So when the viral particles are sitting onto the human cells, the glycoproteins must be cleaved by cathepsin so that this viral particles is uncoated, and that's how it is able to release the genetic material in the human cells. So the researcher, they just tried to block this stage so that cathepsin is not able to cleave the glycoproteins, but at the same time they wanted to be very specific that they don't want to impact the cathepsin, otherwise that will trigger some adverse effects, side effects. But at the same time they wanted to block the entry of the coronavirus within the human cells. So how they proceeded with the lead discovery? As a part of this, they first synthesized the viral and host-derived peptides, and these peptides were labeled so that the fluorescence assay is possible to check. Because if there's cleavage of proteins by cathepsin, it resulted into the emission of light, and that also indicated if your small molecule is able to block the cleavage or not. And to detect this, to perform these assays, they came up with the high throughput screening assay. In that case, the fluorescence assay was used. Dyes were used to label the peptides. And as I said before, if there's a cleavage, which was indicated by increased fluorescence over time, and if there is no increase in fluorescence, in many cases where the cathepsin was not impacting or not binding to the peptide. And for this work, they used the library of around 5,000 compounds, which was commercially available. Even they had to use the pseudotype viruses generated by the cells. And when they tried to determine the IC50 is the inhibitory concentration, which is responsible for the blocking the entry of 50% of viruses within the cells. And to do these studies, the pseudotype viruses were added to the cells in the presence of the different small molecule concentration. After 72 hours, the cells were lysed, and the luciferase expression system was used to check what concentration of small molecules is needed to inhibit the entry of 50% of the viral particles. Naturally, you don't want to have any compound which is cytotoxic, so it's very important you should also be able to determine what is the cytotoxicity of that particular compound, and this is normally done by MTT assay. So this was all about the infection, the status of the vaccines, small molecules, what it means when we want to develop new drugs. And once we have the candidate on the table as a formulation scientist, we must think what are the challenges we have to develop the formulations suitable for preclinical and clinical trials. So let's start with the vaccines. So development of vaccines is really complex, and expensive process because the risk associated is quite high. And so the reason majority of vaccine candidates they fail in preclinical and clinical trials. And as the process is complex, the composition is quite complex. So as a formulation scientist, we should have very good understanding of the factors which can impact the stability of the formulation, because stability is directly linked to the safety and efficacy. When I say safety and efficacy, imagine you have vaccine product where there are any degradation products, unknown antigens, they can trigger some other immunogenic reaction, and naturally then we are dealing with some safety issues. Let's say if the formulation is not properly optimized, you have some significant degradation, you are not able to get the desired efficacy, and that's the reason the desired stability of the vaccine formulation must be attained. And when you think of developing vaccine formulation, especially at the early stage, we need to consider the different factors listed on the slide, notably the biophysical characterization, screening of stabilizers, adjuvant interaction, sterile filtration, container interaction, real-time and axial stability condition. As you can see, all these factors are overlapping between the preclinical phase 1 and phase 2 stage. When you go for submission for phase 1 studies, often regulatory authorities are not stringent. You can still live with some, let's say, ongoing studies. You can commit for additional studies, and some of the studies even you can do phase 2 studies. Typical example, I can give you extractable leachable studies. Sometimes you do have option that you don't have to submit all the data at phase 1. Maybe some of the data you can still submit in phase 2 depending on the type of packaging systems you're using. Let's start with the biophysical characterization. So, in the early phases of the development, it's very important to understand the physical characteristics of the antigen, like its tertiary structure, and we should also know which parameters like pH, buffer species, ionic strength, these are important to retain the folded state of antigen so that no aggregation is taking place. And once we know all these characters, it's very important to maintain the physical stability of antigen, because it is also going to help us for the selection of our favorite excipients. We can also think of using the systematic three-step approach. In that case, antigen is evaluated as a function of pH and temperature, and then we can screen the library of compounds which are helpful to stabilize the antigen and thereby the vaccine formulation. For this purpose, understanding the mechanism of protein aggregation is very, very helpful, so that we know at least which are the factors responsible for protein aggregation. And in order to come with the high throughput screening method, we should be able to detect these aggregates of proteins quite easily. On this slide, I try to list down some of the analytical methods you can think of. So can be used. It is a non-invasive technique, but it's very time consuming. Size exclusion chromatography is very popular, but it needs quite a high quantity of proteins. Other methods like circular dichroism, dynamic light scattering, they are also quite useful, but often these are not available in all the labs. Analytical centrifugation also works quite well, but again, it's very expensive. And none of this method they really provide some high throughput screening. So in that case, you can think of using fluorescence-based protein stability screens. In because in you can use 96 well plate for the fluorescence analysis. In this method, you can think of using fluorescent dyes, and these dyes are able to detect the changes with respect to the protein's folding state. Even there are reports they came up with the time-dependent protein stability assay, which provide the half-life values. So in this case, what they do normally, they prepare a protein samples and it is mixed with the reactive dyes at a fixed temperature, and the fluorescence is measured over a period of time. The half-life corresponds to the time where 50% of the maximum fluorescence is recorded, and this is also indication of protein stability. And this technique can also be used to identify the factors which are responsible for the stabilization of the protein. In addition to the pH evaluation, we can also think of using empirical phase diagram approach, which can help to understand the effect of other variables on protein stability like buffer type or concentration. As you can see from the graph, in this diagram, the different color region they indicate the properties of the antigen. So basically, In the green region, proteins in the native state, when it is changing from green to blue, the unfolding is taking place, and when it is red, the protein aggregation is taking place. So when you are increasing temperature, let's say from 10 degrees to 90 degrees Celsius, you can really see that protein is getting from its native state to aggregation state. Same is the impact of the pH. And this really helps you to check at under which pH and temperature conditions it is possible to keep the protein into the native state. Then evaluation of stabilizers as a, let's say, excipients, because alone pH, ionic strength, or buffer species may not be sufficient enough to stabilize the proteins, especially when you are looking for 36 months of shelf life for your vaccine. So we certainly need a stabilizers. So these stabilizers act by inhibiting the aggregation denatures of the proteins. And selection of stabilizers, when you do, it's very important. They must be from the grass category. They generally recognized as safe category, because then you don't need to discuss with your regulatory authorities regarding the safety of these excipients, because time is always crucial when you want to develop the vaccine as urgent as possible. So these grass traces will certainly help. And especially at early stage, we do not know the mechanism of antigen degradation, so maybe it's very important you want to use the combination of stabilizers which are acting to stabilize the protein by different mechanisms. And to screen the stabilizers, we must know under which condition the protein is getting degraded, so that those stress conditions can be used to stabilize the protein. On this slide, I tried to put the example of different stabilizers we can consider, like amino acids, antioxidants, cyclodextrins, proteins, surfactants, and as I said before, you can also think of using combination. You should have some stress conditions in mind when you want to screen the stabilizers. So in this case, as you see from the from the graph, the protein sample is heated as a at different temperatures, and the excipients are added, and you can see that one excipient is able to stabilize the protein, which was detected by optical density. So when protein is denatured and aggregating naturally, there will be increase in the optical density, and this helps to detect under which conditions, using which excipient the protein is in the stable state. Then the correlation of the real-time and accelerated stability, because at early stage you do not have sufficient time to generate the real-time stability data for 24 to 36 months. So there you can certainly think of generating some accelerated stability data. As an example, if you are 6 months data at 4075, you're allowed to assign a shelf life of 12 months at 2560, which is real-time storage conditions for small molecule. If it is a vaccine compound, often they are stored at 2 to 8 degrees Celsius. And under these conditions, storage at 2560 serves as a stress condition. So having said that, if you're 6 months data at 2560, you can assign a shelf life of 12 months at 2 to 8 degrees Celsius. And when you do such predictions, the Arrhenius equation is very, very important. So that you can check whether it is possible to find any correlation between the real-time and accelerated stability data. Then next point is the adjuvant addition. If you see the conventional data, conventionally they were using just dead, let's say the kill bacteria or viruses as vaccines, so it had sufficient immunogenic potential. But as there was advancement in the purification technologies, we have like very purified vaccines, proteins, and they lack to generate some desired immunogenicity, and that's why the addition of these adjuvants are needed. These adjuvants, they work by enhancing the immunogenic potential of the viruses. So in this case, if you see the choice of adjuvants, the majority of the commercial vaccine, they are using aluminum salts as adjuvants. There are also other adjuvants reported like squalene-based O/W emulsion. When you have like toll-like receptor agonists, saponins as an example of adjuvants. So in that case, it's very important to verify the adjuvant is not interacting with the vaccine or with the protein. Sterile filtration is very important, let's say the process, because at the end you want to develop a sterile product. So these vaccines, they must be produced under aseptic area. At the end, if it is possible, sterile filtration is needed, especially for the solution based formulation. But let's say if your vaccine which is having adjuvant in the form of aluminum particles, so they cannot be filtered, so in that case it's very important that entire operation must be done under aseptic area. The filters you are using, like PES, cellulose acetate, for PBDF, they must not react with the vaccines. At the same time, the vaccine formulation must not block these filters, and that's why we do the filter validation studies during the development of parental products. You see on this slide on the left, you have the graph where the viral particles were passed through the different filters, and in case of the cellulose acetate filters, the clogging was happening, and that's the reason there was significant increase in the pressure. This was followed by PVDF and PES. So PES membrane was very less prone to the blockage, and that's why the increase of pressure was quite significantly delayed. This was also confirmed by additional experiments where the vaccine formulation was filtered through the different filters, and the percent loss on filter was the least when the PES filter was used. So these studies are very helpful to select a particular filter because you don't want any interaction of your formulation with the filter membranes. Once your vaccine formulation is ready, you certainly should have the analytical specification or the different specifications in place so that the product is ready for the release. So general properties you take into account there like appearance, water content, which is important for lyophilized product. Then pH and osmolality must be acceptable physiologically. When the lyophilized product is reconstituted, its appearance should be acceptable. Then the volume you are able to extract should be within the limits. Uniformity mass is also very important. You must identify the target protein, endotoxin content, as well as sterility. So this was all about vaccine formulation development. Let's start discussing small molecules. So when you have small molecule on the table, you do have a choice for route of administration, oral followed by parenteral. Oral route is certainly very well preferred, but as we discussed before, compounds like remdesivir, it is given by IV route. So naturally, you need to select the parenteral options there. So in that case, when you go for parenteral formulations, you do have a choice between intramuscular, subcutaneous, and IV formulation. But if the dose is very high, you cannot really think of intramuscular or subcutaneous formulation because the volume of injection is hardly between 2 to 5 ml, which cannot really accommodate all the dose. Certainly, you need to think of IV infusions. And when you think of IV infusions, the poor solubility of these compounds may be one of the issues because if the compound is showing very very low solubility, it will be very difficult to get that compound in the solution form, especially where the dose is very very high. Maybe you have options of using the nanosizing if you are not able to solubilize all the drug, but then certainly you need to take into consideration the particle size needed for the nanosizing. And just to get some idea, the 40% of marketed drugs, they represent the compounds from the low solubility class. If you see the drugs in pipeline, around 90% of those molecules are from the class of low soluble compounds. So the critical quality attributes you need to consider while developing the IV formulation, they are like excipient selection. So to develop an IV formulation, you need excipients such as solubilizers, buffering agents, antioxidants, and tonicity agents. The pH must be physiologically acceptable, that is around 7.4, but often it is not possible due to the low solubility or stability of your compound. So majority of licensed products, you see they have the pH between 3 to 9. Above pH 9, there are incidences of tissue necrosis. Below pH 3, it may cause some pain like side effects. Osmolality specifications, often it is between 280 to 290 milliosmoles per liter, but that's often not the case. When you see some hyper tonic solutions having osmolality even up to 600 to 900 milliosmoles per liter, depending on the volume of infusion. But the hypotonic solutions should be avoided because they are known to cause the hemolysis. So, what are the possible strategies we do have to formulate the poorly soluble drug? So you can think of pH modification, use of co-solvents or surfactants, lipid-based formulation like microemulsions, interlipid emulsions, liposome, mixed mixes, use of cyclodextrin nanosuspension. But trust me, if none of this option is working for your target molecule, so it's better to say that the molecule is not really suited for a IV infusion or IV formulation. It's better to do any structural modification. Maybe you should come with some product type of concept rather than spending time on the formulation research. So when you think of pH modification, basically you are just trying to take the advantage of ionization of that particular compound. So around 70% of drugs on the market, they show some ionizable groups. So pH dependent solubility can be explored there, and it also helps to come up with the salts. For example, if you have like weakly acidic compound, you may come with some basic salts. If you have like weakly basic compound, you can come with some acidic salts, and that's how you want to increase the solubility of those compounds. Examples from the commercial compounds, you can take the ciprofloxacin, which is a broad spectrum antibacterial compound. The commercial formulation contains the lactic acid as a pH modifier, and that's how they increase the solubility of the compound. Another example you can see of aspirin. This drug is basically invented at aspirin by Uppertal, but this drug is still being used for a lot of other indication. And recently they tried to use this compound for relieving the migraine pain. And the trials they did in comparison with sumatriptan. And as the dose needed was quite high, they came up with the DL-lysine salt of aspirin, which was highly soluble. So advantage of this salt compared to sumatriptan was it was very well tolerated, though it was less efficacious, but tolerance was very good. The drawback of this approach is maybe to in view precipitation, because rapid shift of the pH taking place. Imagine you have formulation with pH 4, which is injected in vivo where pH is 7.4. There is a rapid pH shift, and that's where the rapid precipitation of the API is taking place. And you would like to avoid this, and that's the reason you should generate sufficient in vitro data to make sure that the in vivo precipitation is not taking place. The next strategy is use of co-solvents and surfactants. So in that case, basically we are just trying to use the co-solvents and surfactants to solubilize the compounds, and they act by changing the dielectric constant of the drugs, and that's how they facilitate the solution of non-polar drugs. Often the co-solvent they are used in combination surfactants, pH modifier, so that this combination is able to increase the solubility. The most debated formulation from this class is the Axial formulation, which contains 51% chromophore. So as you all know, the pre-treatment of antihistamines is very important because chromophore is known to induce some hypersensitivity reaction, and due to that reason, other players were working and they came up with the chromophore EL-free formulations. And the commercial success of this approach is Abroxane, which is a nanosuspension based formulation of paclitaxel. Then another example from this class, docetaxel, the commercial formulation of this compound is taxotere, which contains 54% of polysorbate. Again, very high concentration of this polysorbate, it leads to hypersensitivity type of reaction, and that's where other places like Sandoz Hospital they use 5,5B2 approach. They replace 2080 with PEC 300, and that's how they were able to reduce the incidence of hypersensitivity reactions. But there are disadvantages of this approach as well, because often the poor tolerability is shown for this formulation due to the very high concentration of co-solvent and surfactants. Uncontrolled precipitation is taking place because once these formulations are administered, uh, miscibility of this co-solvent surfactants with plasma leads to rapid precipitation of the drug, and this is something to be checked by conducting some in vitro test. Cyclodextrins is also very interesting example. So basically, these are cyclic molecules with hydrophobic cavity. So hydrophobic part of the API is sitting into these cavities, and the resulting complex is soluble. Advantage of this approach: it's very easy process. You can work with very small quantities of API, and that's why this approach is very popular for parental route. The most debated the cyclodextrin these days is Captisol. I came up across very interesting example, the Westitane is compound, which is being investigated as a potential treatment for post-operative nausea and vomiting. But the major disadvantage of this investigational compound was its surfactant-like properties, which leads to hemolysis. And this mannitol-based formation, which they had, was to be induced very slowly, was to be infused very slowly, that so that they were able to control the hemolytic potential of this compound in the preclinical studies. Then they came up with the captosol-based formulation. So when they tried to bind the drug to the captisol, there was very less amount of free drug into the plasma, and that's how they tried to control the hemolysis. And with the captisol containing formulation, they were able to infuse around 50 milligram of the compound over 30 seconds, which was not the case for mineral based formulation. Then we have a choice of lipid based formulation. This approach is very, very important for the drugs with high log P. So in this case, stability of EP in contact with lipid is very, very important because the residual oxidizing impurities in lipids, they may lead to degradation of the drug. Concentration of surfactant should be carefully considered because, just we discussed, it may lead to some tolerance issues. As lipids and surfactants, they cannot induce the desired osmolality, so addition of tonicity agent should be considered. Microemulsions is one of the very famous example from this class. Basically, just mixture of oil, surfactants, co-surfactant, water, which leads to clear emulsion at the end. The energy is needed not very high, and this can be used to incorporate the drug. Let's say with the loading of 5 to 10%. Really depends on the solubility, and typically these formulations have lipid drop is in the range of 10 to 20%. Commercial success from this class is propofol injection, which was earlier chromophore based formulation, which was later on ethanol based formulation, and finally they came up with the micromulsion based formulation, which is quite highly tolerated and safety profile looks quite good. Mixed match is very interesting example. Even I tried to use it during my times at Farm Swiss. There was a company called eFares. They had this ready-to-use kit. So basically, they used to provide the ready-to-use mixed micelle, which was aqueous buffer containing phospholipid. So we simply dissolved the drug in ethanol, and this ethanolic solution was mixed with this mixed micellar solution, and at the end we were able to obtain a clear solution. And this formulation was very readily available for any preclinical studies, so that we don't have to spend a lot of time to develop IV formulation of investigational compound. Then we do have a choice of also liposomes. So basically, these are spherical structures containing aqueous cores encapsulated by natural or synthetic phospholipids. The commercial success from this class is doxorubicin, which is an anti-cancer drug. The major disadvantage of this drug is that it binds to the heart protein called cardiolipin, and this may even lead to irreversible congestive failure, and that's why this drug is not very well tolerated. So initially they tried to come up with the negatively charged liposomes, but these liposomes were not quite successful because they were cleared by reticular endothelial system. Then they tried to use the loading approach. So basically they use the remote loading, which improved drug loading efficiency, and even the formulation was showing quite good stability. And that's how it resulted into the discovery of two products, myosit and doxil, where the pH is loaded by pH gradient method. As you see from the graph, there was a significant increase in the bioavailability from myosit when you compare with the free drug. Even the half-life was significantly increased. And in terms of side effects, if you see cardiac events, mucositis, nausea, the liposomal formulation was very well tolerated compared to the free drug. Then another example you have from this class of the formulation is intralipid emulsion. So basically, these are used as a part of the total parental nutrition, which serve as a source of energy for the elderly patients. So these are made up of lipids, emulsifiers, and aqueous phase. Advantages that there is less pain at the site of injection, less toxicity, the stability is quite good. However, when you see the drawbacks of this approach, often the drugs are not showing quite high solubility into the lipids, and that's why the loading is not that high. You may see some instability issues, especially when your drug is not stable with the lipids. Also, the choice of emulsifiers is quite limited. The process looks also quite simple. Drug is dissolved in oily phase. It is mixed with the aqueous phase. It contains surfactants, which is subjected to the high shear mixture. And when you have coarse emulsion, it is subjected to the microfluid digestion. And at the end, you have fine emulsion, which is subjected to core filtration, and because you want to have the particle size below 5 micron, acceptable for IV infusion, and which is subject to the sterilization. And then the last class of strategy for improving the solubility of small compounds, the nanosuspension. So this technique is quite popular to improve the bioavailability of the drugs administered by oral route. But nowadays, it's also being considered for the parenteral route. So now then, basically, the nanoparticles are produced by bottom and top-down approach. In case of bottom-up technology, the drug is dissolved in a solvent, where the controlled precipitation is done by adding the anti-solvent. Then other options are using of high pressure homogenization as well as media milling to obtain the nanoparticles. And Abraxane is a commercial success from this class, which is a nano suspension based formulation of paclitaxel, where they have drug loading of 10%, you have 90% albumin. Their particle size is in the range of 113 nanometer. And as you can see, the maximum tolerated dose from this nano suspension formulation was very high compared to the taxol formulation. Then one should also consider the admix compatibility studies as well as extractable leachable studies for the parental formulation, because you don't want to see an interaction of your formulation with the infusion bags, the infusion line, as well as the syringes. So neither your drug should get absorbed onto this bags or packaging materials, and you also don't want any material from this any component from these materials should get into your formulation, which may result into some tox issues. And that's why the admix compatibility studies, extractable leachable studies should be planned with the final formulation, so that this data is available at the time of submission. So with this, I would like to conclude today's talk. So development time for the new drug or vaccine should be taken into account when we are considering the severity of the corona disease. The modeling studies should help to select the repurposing of the drugs against corona. Often the combination of drugs should help to target the multiple pathways. Vaccine formulation, they represent quite complex composition and process. Hence, we need to be very careful when we consider the formulation components. The formulation of poorly soluble drugs, it's a major challenge to deliver by IV route. And there the selection of excipients is very important. Some of the technologies, they are quite easy, like mixed masters, which we discussed at early stage. IP perspective should be considered, which gives us freedom to operate, especially for Captisol, which is a patented excipient. If we want to use it, we must have some licensing deals with Sidex. We must consider some in vitro tools to make sure that in vivo precipitation is not happening, and admix compatibility data extractables and leachable studies must be performed in advance, so that we are not getting into any incompatibility issues. So with that, I would like to conclude my today's talk. Thank you for your attention.
Hello. Yeah, excellent talk, sir. We are very thankful to you for this excellent talk, and we wholeheartedly congratulate you on behalf of our university for holding this talk. I have been listing out the questions. There have been a long list of the questions, and I would like to take up a few questions. The first question being a general question: what is the difference between a vaccine and plasma therapy?
So basically, when I say vaccine, you do have in protein component, as we discussed before, and this protein component is representing to the coronavirus's protein, so that there are chances to create an immune response. So it's very important there that whatever component present in the vaccine, it should have some resemblance to the coronavirus's protein. And when we say plasma therapy, so the basis there is that Ima- imagine any patient who was infected with corona. Naturally, there was some immunity generated in that patient, so that plasma must be containing antibodies against corona, but those antibodies were generated with that human. It has nothing to do with the viral proteins materials. So this is the basic difference. The plasma containing antibodies from the humans, and in the vaccine you do have the proteins which are resembling to the coronavirus proteins.
Sir, the next question is in the same line. When we talk of convalescent plasma therapy, how do you decide the dose?
So basically, this is also question of the titer values. The plasma must be containing antibodies against corona, and then we should know the titer values of those antibodies into the plasma. And when you try to take the swabs from the corona infected patients, so by using this RT-PCR, you try to get some idea what are the titer values in that particular patient, and this is basically the titration against the antigen antibody, and that gives you some clue about the, let's say, the dose of the plasma therapy you need, because the plasma is containing typical antibodies.
Sir, the next question is there are different strains of corona. So, when you think of a vaccine development, do you think that a vaccine would be developed with all the strains, or how? I mean, would you be combining the two strains or three strains, or would it be a one strain which would be effective against all the? The strains.
I mean, when you want, when someone is trying to developing a vaccine, the genetic sequence of that virus, that particular virus, is taken into consideration. So at that moment, you do have one particular genetic sequence into the consideration. So maybe at that time, it's very difficult to predict if it is going to act against all the different strains. So during design, it's very difficult. But when you go for testing, then you do have the possibility. Even in small molecules, we do develop antiviral drugs. So even for small molecules, when we do testing, we try to screen as many strains as possible to make sure that you do have the drug which is acting against all different strains. Maybe there are some sensitive, maybe there are some resistant strains. So that data is only available when you do actual testing. At the time of design, it's very difficult to predict.
Right. Sir, next question is, can binase, which is a RNase, can be applicable to control viruses?
Sorry, can you repeat that question?
Can RNase, binase is RNase, can it be used to control such type of viruses?
Because we discussed the mechanism action of different drugs. So I mean, let's say if you that particular drug which you are discussing, if it is able to block the entry of viruses, whether it's able to block the replication of the RNA into the human cells, if it's able to do that, then of course we can use that. But for that, you certainly need some experimental data. Theoretically, it will be very difficult.
Uh, you are talking about there are different mechanisms by which these antiviral drugs act. So, can you suggest to the participants that which would be the best mechanism, or which the drug would be most effective?
So I mean, we discussed during the talk. Remdesivir was one of the drug we discussed that which is basically inhibiting the viral replication. When we discussed favipiravir, which was inhibiting the replication of the viral genetic material into the proteins. So these mechanisms are quite helpful. At least the data is showing. And when you think about vaccines they are basically triggering some immunogenic response, so that this T helper cells, the B cells we discussed, they are being let's say activated, and that's how they are trying to target the coronavirus.
Sir, one more question. Anything you would like to throw light on herd immunity?
So can you elaborate that? What do you mean by herd
So,
immunity?
herd immunity is like when the contagious disease spreads across the immunity, across the population, then herd immunity develops. So,
I mean, if you see, even you see the statistics, the immune response is big different with the elderly patient, the young patient, even the pediatric patient. So it also depends on the age of the patient, how its status. So basically, in case of diabetic patients, the patients having the cardiovascular issues, the immune response is not that great, but in the healthy patients, immunity is different. So something like community immunity will be something little complex or little difficult to predict in this case. It will be really depending on that particular individual.
sir, one last question. Uh, there are a compound generally when it comes to the clinic, it takes around 15 to 20 years. But
10 to
in this
15. Mhm.
Yeah, but in these times of difficulties, do you think that the regulators should take this on a fast scale, or how long would it take to develop the vaccine and
Yeah,
clear the regulatory hurdles?
right, right. So, I mean, that's the case I was trying to tell you. I mean, in these cases, the regulatory authorities they are quite flexible. So, when they allow you to run your phase one studies quite early, and they will even allow you to do some preclinical studies in parallel. Often, they allow you to do some randomized trials. Often, the availability of patients can be made very, very easily. You can even commit a lot of other studies in parallel. So, the time of development can be really reduced. The biggest challenge I see is getting number of patients there. And running clinical trial is not that easy as we think, because you need to restrict that protocol. You need to really comply with that particular protocol. You need to see what is the outcome of that clinical trial, and that is something time consuming. But at the end of the day, the regulatory authorities will still like to see whether your drug or vaccine is still acting. As we saw in case of remdesivir, basically did not show any difference between control group and remdesivir. So under this condition, based on what they will approve the drug.
Right. Thanks a lot, sir. Now, on behalf of our university, I would like to thank all the participants and the speaker for this exciting talk and enlightening all of us.
Thank you. Thank you very much for giving me this opportunity. I really enjoyed this interaction. Thanks a lot.
Thanks, sir. All of you, I request all the participants to stay home, stay safe.
Thank you.
Thank you.
Bye.